I have to try and peel away good sizzle from good sausage. Is there a there, or is it sizzle? And so I definitely go into the literature, the experimental study design on all engagements like that, just to run my head through the experimental execution, gaps in thinking, technical capability, overreach, under delivery, imprecision.
Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe. Hello and welcome to BioTalk Unzipped, where we unzip, unlock, and uncover the stories behind medical progress. I'm your host, Gregory Austin, a biotech science enthusiast. Along with me from North Carolina is my excellent co-host, Dr. Chad Briscoe, a biotech scientific expert. And we publish BioTalk Unzipped twice a month and bring experts from different corners of the pharmaceutical, biotech, or med device worlds to share their innovations, insights, career journeys, and personal stories to the show. On this, episode 8, we continue our conversation with Dr. Russell Grant.
What does Russ Grant's brain do when he sees a groundbreaking publication? Maybe it's from a group you don't know, or you see a company like Theranos come out with a technology that's like, those of us in the industry go, if we could do it, that'd be phenomenal. But it really makes you skeptical. But at the same time, you don't want to discount something that's really groundbreaking. How does Russ Grant's brain process those things?
So that is a third rail, but I'll take a gentle swing at that one. The first thing that I personally did was, when I was made aware of something like that, was to go to the literature base for all of the people that I knew that were at the company, to try and piece together what it is. There's a lot of stealth mode, obviously, in early startups, and we're trying to get some publicity, but in stealth mode. That's not just that company, that's many companies. So I went to PhD thesis, book chapters, a lot of literature that was, to my understanding, the foundation of the scientists that were working there at the time, to try and figure out what it was, or what these companies sort of have as the magic combination of technologies that sort of makes it work. Now, anecdotally and practically, what happened was we recentered all of our microsampling efforts with, as we've now seen evolution from blood spots to blood plasma separation to liquid microneedle collectibles. All of that work moved from a couple of the laboratories that were at LabCorp underneath me. So the knock-on effect was I got microsampling to try and see if we could anchor it properly with, high-end analytical technologies, and bring that technology to bear as the market, the business model makes sense and made sense. COVID is another perfect example of that. But what it forced, forced me to step up and own that and still do a big part of microsampling technology development, assay development, and clinical utilization. So I'm going to leave it at that. I don't want to get into I think we all know what happened there. But the model was good. But generally, what do I have to do? I have to try and peel away good sizzle from good sausage. Is there a there, or is it sizzle? And so I definitely go into the literature, the experimental study design on all engagements like that, just to run my head through the experimental execution, gaps in thinking, technical capability, overreach, under delivery, imprecision. One of the things that always tends to stop a lot of, and this is through my general conversations and also engagement, I'd like to see a well executed validation report for this particular assay, please. And we submit to regulatory bodies, so I know what, and I've done it in the past through GLP. I know what a quality validation report looks like, but there's a tension there to generate buzz, to generate serious funding, to move these things forward with the intention that perhaps they or a partner will engage and do the boring hard yards. I kind of do the boring hard yards. I kind of like it. I mean, I'm not a fan of the 80% documentation, 20% wet work that my GLP world became. It's quite the flip, but yeah, I think I'd say definitely go back to all of the literature, the foundational science, and just check the assumptions that were made, and then educate back. Have you thought about this experiment? What about that? You're putting these liquids in as an example, a small microtube with a bit of gel in it. Have you figured whether the analyte adsorbs into the gel? Because the liquid to gel ratio is different. Does something come out of the gel? Do you even know? Just probing questions of what ifs.
How do you even control? How do you even control how much liquid goes in? Right. That's where the change is. I think we were just having that conversation today about an assay with a customer, because it's a swab assay. I'll leave it at that. Right. So how do you even know how much is getting into the swab? I mean, there's these simple questions that they're simple questions with very challenging answers, and we've literally been discussing this for weeks. And analyzing it, and it's incredible to think about that, but it's true, and you understand.
Yeah, I think there's two things that I always think about. How can I normalize? Well, how can I get to a amount per mil, per cell, per mass? How can I normalize a collection that's not subaliquoting with a known volume or a known amount? How do I normalize is question one. And then question two, things like how variable a liquid can I allow for it? For instance, for a microsample collection, a liquid or a size of a blood spot, how much latitude in collection do I have through, in vitro, in lab validation studies, and then you have a monitoring process to say within these Goldilocks zone, good, outside, not good. And you support that with data. More often than not, I whiteboard experiments to try and technically get at the answer to these what-if scenarios in a robust, repeatable, but scientifically defensible manner. And I find myself whiteboarding that all the time with my team, because we're always at the edge of, how do I even do that? How can I prove if the swab was fully saturated? What have I got to do? What can I do when we have access to kind of all the tools in the world in a diagnostic lab? So I think about that all the time, and I try and figure that out. If quantity is what you want, and I'd say almost my entire career has been based around quantification.
Sure.
That's really where I think I've tried to be above average.
Excellent. Well, again, gentlemen, it's time for new news.
New news.
So in this recent article that published on, I think it was January 30th through Fierce Biotech, Elon Musk's company Neuralink started its first FDA-approved clinical trial in humans, in which a patient received a neural implant on January 28th, and the initial results showed some promising neuron spike detection. So the overall goal of this technology is a kind of a long-term, is to help maybe paralytics walk and communicate better, and maybe even the blind see. And so this implant they put into this human has over 1,000 electrodes delivered to a very specific region of the brain to record this neurological activity and then transfer it, I assume via Bluetooth, to a connected app dubbed telepathy. And so what this is supposed to enable people to do who can't move their body very well is to control their phone or computer just by thinking. Kind of crazy, right? What are some of the pros and cons of this budding technology?
So I did read that story, and it reminds me of that old Clint Eastwood movie where he controlled that airplane with his brain. I don't know if you remember that one from the 80s. I loved that movie.
That was a cool movie.
It'll come back to me. I'm fortunate enough again to be working in neurology. A number of years ago, somebody sort of put a bug in my ear that neurology and neuroscience is the cancer of the 21st century. As we become better at solving for certain cancers, maybe the brain is the next sort of sphere of analytical and biological understanding. And we're of course seeing that with the therapeutics that are coming along. I've been fortunate enough to work on scaling up and now launching tests here at LabCorp for A beta peptides, NFL, PTA-181. We have others obviously to come. So the biomarkers in plasma, not in CSF, being part of that journey. So you have accessibility to the matrix to make these sort of informed decisions is where we're in that moment. I think of a computer or a human computer or systems hybrid as an incredible opportunity and fraught with obviously obvious risks of health, safety, efficacy, unanticipated consequences. I think there's a wonderful Black Mirror episode where a woman, she unfortunately passes away in the episode, and she has her whole persona put into a small teddy bear to grow up with her child. Well, how do you update that kind of technology following an invasive surgery? And do you want to be early? Do you want to be middle and late? So anything we can do, I think, to challenge and tackle the diseases and issues with neurology, I think is going to be fascinating and get lots of buzz, particularly with Elon Musk's name associated with it. But I think the counter is it's highly risky, and we still don't really understand all of the biochemistry of tau pathology, and we've been studying that molecule for 30 odd years. So for me, I'm very much on the side of, I know we have to have advancement. I'd like to do no harm. I'd like to do it in the least invasive way, but understand that things like that are obviously part of what a human future could well be, with people like obviously Elon driving it. Bit worrying to me. I don't think I'd be an early adopter. No, I don't think that would be me.
But if you were desperate, right? If you were someone with a
Sure.
limitation to what you could do because of not having control for whatever reason, you thought you could fix it, then that might change the perspective.
Naturally, everybody's perception is their own unique journey. I couldn't comment on people's decision, but it's
Sure.
it's a promise and an opportunity for people. And of course, if it's safe and ideally effective, then more power to any companies doing that.
Yeah, it's going to be a longitudinal study to see over time how this actually works or doesn't work, and we'll have to see how it develops, if it develops.
I think it will, right? It's just a question of where it goes, and whether it's today with this technology or some technology 25 years in the future, 50 years in the future. Yeah,
right.
Naturally, I haven't seen a tricorder yet, but by gosh, I want one when they're made, right?
Right.
Yeah.
I haven't seen a neuralizer yet, but I wouldn't mind one of them once or twice.
Yeah, it could have been useful back. Back in the younger days. Had a few maybe neuralize yourself. Forget the night.
Right. Speaking for yourself there, Chad, not me. Thank you, mate.
I know. I started going down a rabbit hole with that one, and I couldn't quite pull myself out.
Well, great, Russ. I was doing some research on you, and some of your contributions to TAMS, the Triangle Area Mass Spec group, has been dubbed as legendary and seemingly limitless. What do you attribute this to, and what drives you in that area?
A lucky or unlucky conversation at a booth at ASMS, just as I'd moved from North Carolina. So LabCorp asked me to move after the acquisition. And a couple of people said, you're known, you're coming to the area, would you mind taking this on? I said, sure, but I would like to scope it the way I would like it to happen. We had to sit down. I said, look, guys, we have so many people in the North Carolina community, where I also am aware Chad is, that teach at conferences, give wonderful short courses. Let's do a summer teaching series for free. Just do that. Cool. Got that. But what I'd like to do is to invite in world-class speakers to give a main plenary talk, but I want grad students to give an intro talk. So I want grad students to have a connection to an external professor or expert or whatever. I would like this meeting to meet every month of the year except the ASMS month. I want the grad students to get window access, because if we don't build a solid foundation and trunk that follow us, then we haven't done our job right. So for me, it was about empowering the students, giving them shop window or opportunity to be in front of the local scientists, but also to engage with an external scientist for a lifelong career of camaraderie. and just a passion for teaching and trying to take whatever it is I've learned and give it freely so that it doesn't die inside my head. And so they fortuitously, the guys said, yeah, okay, we're down. So I did it for 6 years and then found some other wonderful scientists to carry on. It was a 6-year window for anybody who's taken it on. Since then in 2-year chunks, but I chaired for 6 years. But I just wanted to make sure that we vicariously and altruistically gave back to our junior scientists the opportunity to have access to us, to not look at us as scary big names, as I looked at the people I first met at ASMS in '95. I'm stood next to Dick Smith and Hillenkamp. I'm given a poster. I was petrified because these are legends. I'm a nobody. Let's break that down and let's give back to these budding scientists and give them every opportunity to take advantage of our vasculature in the network, in our networks, our hypervascularization. I'm gonna get that word right. Our vascularized nature as professors and seniors in the industry, and just give these young men and women an opportunity to go wherever they want. So it was about giving back, but it was with the knowledge that I would maybe get first access to a couple of local students that were exceptional.
Sure.
I could probably hire a couple, and I did. So
Yeah.
there was a little bit of this worked out really well for me as well. But great for our local community. It fosters really strong networks between academia, industry, and our manufacturing partners. And what drives it all? Just a passion to, you got to care. You got to give something back, I think, and we're fortunate enough to have that as a community. But I took that and just ran with it.
That's
Thank you for the
brilliant.
very kind words.
Yeah,
Yeah, of
yeah.
course.
Well, obviously, it's having an impact because I don't know if that was a former student in that process who wrote that comment, but it definitely made a difference, and it's so much more um, impactful to have them being engaged in that kind of way instead of just sort of passively hearing and maybe getting a handshake, and really encouraging that type of networking. As you know, it's so important, especially in this field, because it's a small community, really.
It
So
is.
congratulations.
Well, thanks. There's a funny thing though, because I have a bit of a reputation of being that guy to go to the microphone and ask that question, make you think about what you put up on screen. And Chad's nodding. I have that reputation. It's sort of funny. There's me being the president, trying to engage and try and and I think we've done a fabulous job. Whenever I don't go to a TAMS, the student who was speaking that night will email me like, I was ready for your question. I'm disappointed you weren't there to make me think about what I'm showing and make me think deeper. I, I'm a teddy bear inside. I really care about scientific learning and teaching and the evolution of our next generations. I really care about that. We should never walk out of our careers with stuff in our head that we've not populated in somebody else's head, and the passion that we have infected into them. We shouldn't ever do that. That's
But
wasteful.
so having seen you do that, countless times, Russ,
Hmm.
come up and ask the question. First of all, when you don't, it is missed. It's not just missed by the person expecting it, but the audience is looking for where's that fantastic Russ Grant question. But the other thing I've seen you do is you ask a really tough question. It makes them think, but then you're not trying to make the person look bad on stage or demean it, and then you'll assess who that is, and you'll kind of step them through it, right? Almost help them find the answer in a way that they learn, that everybody in the audience learns, and doesn't leave them feeling like, man, I just did my first podium presentation for a couple hundred people and I can't do this again. They come out of it, I think, again, like you said, feeling like, wow, I learned. I'm going to be better next time. So
That is,
that's a skill. That's an amazing skill, actually.
well, very gracious of you to say that. Not everybody sees it that way, but certainly the intent is to try and use, again, I think a podium presentation, you're the reviewer in live format. It's not a written review comments back to a
Yeah.
paper. So some of that you have to gauge, okay, I'm going to have to take this down a few steps to make this person get an aha. Oh, yeah, I got something, and or correct something that perhaps has been overstated to the audience when we
Sure.
came back to repeatability. Probably one thing that you may or may not be aware, I'm also an adjunct professor at UNC at the Department of Chemistry, and I have been for
Okay.
about 9 years. And every Thursday I give up 3-4 hours of my life for free to work with grad students, and I focus very hard on scientific method, scientific messaging, and precision in execution, but depth in understanding. So for me, it's a passion to get people to not take an easy answer with canned workflows on tools that we now have access to, but actually think through the foundational hypothesis, but the assumptions that are made when they put some data up on a screen that the latest and greatest instrument has provided. No, think through it. Have you seen the data and what it's actually telling you, and did you do the experiment correctly or not? And I always try to combat the necessary simplification of the workflow and the instrumentation, which is required to obviously permeate mass spectrometry, for instance, across multiple disciplines, but always try and get people to anchor back to, I need you to look, well, you said this, can you just look at that data? Are you sure that's what that says? And get them to just check themselves again.
Mm-hmm.
That's the general intent. Not everybody reads it that way, and I'll obviously have sidebar conversations with them afterwards. But it's to try and honour I think the scientific underpinning of what people are trying to do, and add a little bit more thoughtfulness into their work as they move forward. And I think that's the genesis. I was ready for you. I was ready for another question. I wanted you in the audience to ask something to make me think harder about what I'm doing. Ian Wilson taught me that. Ian Wilson taught me that.
It reminds me, we're not bumping into each other at meetings the way we had in the past, but when you would give talks and I'd see, and I won't use last names, but I'd see Kevin or Andy or number of other folks that they're going to poke at Russ on this one. I'm looking forward to seeing where this goes. And I know you welcome that, right? And you welcome the challenge, but there's only a handful of people who really, because you are so thorough, there's only a handful of really top-notch people in our field that can do that to you. I've probably tried unsuccessfully, but there's a few guys who've been able to poke at you.
That is really nice. Yeah, that's really nice. My father gave me one thing once, not from this field, not a scientist, and but he couched it really well. And he said, Russ, could he see me present just a couple of times? He said, when you present, you're only looking for one thing. And we've done this when we've had posters and people like, Rick King has walked past and he sort of scanned and you get, and Kev Bateman, legends, absolutely incredible scientists. People like Russ as well. Russ Weiner was on one of the earlier podcasts. My dad says, all you're looking for the 4 or 5 people in the room that are about the best there is, you're looking for, he called it the nod of the artisan. You'll show something and you'll just get this almost imperceptible nod. You're like, yep. He said, that's it, Russ. That's all you look for as the validation of what you've done scientifically and composed is the nod of the artisan. And it's actually really true. And to even think that I'm approaching the quality of those scientists is humbling because they're incredible. But I've tried, I've tried to be that. The nod of the artisan. It was a wonderful, sort of thoughtful moment from my father.
I love that as well. And I guess I'll tell you, I feel like I've gotten it from you a couple of times and I'm feeling better now. I feel like, wow. Yeah. Your talk at the sample preparation workshop for ASMS in 2016 was phenomenal. I loved it. I absolutely loved that. So I was happy to share that with you. Your talk was great. It was a good time.
I'm still thinking about how to do bioanalysis of bone, but your talk was awesome. I think you were doing teeth or something. It was great. I enjoyed it.
Yeah.
Excellent. And going back to the grad students for a minute, what do you see as some of maybe the biggest concerns or obstacles for them today that might have been different than maybe 10 or 20 years ago?
I have a personal opinion of what I think grad school is for, and I think it's foundational. I think it's learning the scientific methodology or the hypothesis generation methodology, but I don't think it's the right vehicle for translational work. I think it's better. I am a big believer of academia for fundamentals, for risky fundamental sort of, hypothesis generating questions. So I see a lot of a push towards application for funding, which again concerns me. I think people like myself and Chad and others that I've named are incredible at honing a workflow or a process to generate highly precise, highly accurate, or low bias data. So I think there's a push towards application, less on fundamentals. And I'm still a believer that if you don't, as I said, I spent 6 months, 5-6 hours a day for 6 months in the literature, reading the work of Archer Martin and J.J. Thompson, Van Diemter, and Cal Giddings. I try to get deep into all of this foundational understanding, so I knew roughly what the guardrails are of scientifically credible data. It'd be nice to see a little bit more foundational, fundamental work, I think. In terms of the science, I'm lucky to work at UNC. It's an incredible program. The students coming through their PhD programs, they never have any issues. It's a top quality school, has been, forever. So I'm lucky enough to work with the cream of the cream, and try and add a little bit of something to them. I think probably one thing that I would continue to espouse to any grad student is try and be brilliant at something. Not good, not great. Find out what it is that you can be brilliant at. And I do mean world-class brilliant. Hopefully it's something that's got legs and it's got many decades of growth for you, but find something to be brilliant and be brilliant at it, truly brilliant. And your career will look after itself. Integrity in data generation, be honest, be collegial, tell the truth, do quality work, but be brilliant at something. All those things sort of amalgamate. Just be brilliant. Your career will look after yourself, or itself.
Absolutely.
Be brilliant.
That's good. That's so good. Russ, tell me about a time when one of your tests or innovations led to something truly unexpected.
Well, again, testosterone. I was asked, could I develop a testosterone assay that was 50-fold lower than anything that had ever been done? I'm like, I have no idea. I said, but what we'll do is we will ask them to make an internal standard, because I wanted a better labeled internal standard. We'll own the rights to that. This is when I was at Esoterics, and I'll see what I can do. I have no idea. So I went from 20 picogram per mil down to 300 to 200 femtogram per mil. And femtogram per mil, we had to have women of females, do preparation because of the sweat in men, even gloved up, and we couldn't have men prepare the tests. So it forced me to go nearly 100 or 50-fold lower, which forced me to do a couple of other things. Really polish my two-dimensional LC separation skills, figure out how to create matrices that behave like human matrix, but I couldn't get clean enough charcoal stripped matrix. I couldn't. So then I had to get into, how do I manufacture a calibration matrix that's clean enough to go down, not 50-fold, but actually 250-fold for the 20% LOQ contamination level. How do I make that? How do I store that? As we're getting into these dilute samples, how do I deal with absorptive loss and losses? So it forced me to do two things. It forced me to really clean up how I, or the assay was executed, clean up the laboratory, because 50-fold lower is 100-fold more interferences, right? It forced me to polish off two-dimensional separations. The kind of the funny thing is I have a habit of screening many columns to try and deconvolve selectivity in the first dimension from separation sensitivity in the second dimension. So you decouple those two competing variables. The column that worked, and I'd screened about 60 or 70 columns, and I handed it off to a colleague to screen a few more. We ordered a column from Agilent. It came in, it was the wrong dimension, the wrong particle size, the wrong pore size, the wrong internal diameter, and it worked. It resolved all of those interferences. I'm like, we ran it anyway. It was completely the wrong column, and it just magically, this resolving separation in the first dimension came out. I've never published it. I've never presented it. I'm never going to give it away, because you'd never think to go there. So value of serendipity.
Excellent.
I love
Excellent.
that.
It also, funnily enough, I also made me realize that there's a way to make it inside. So that was a piece of work for androgen suppressing therapies. The salesperson at the time marketed that, and we actually got clinical studies for many major pharmaceutical companies based on that assay that we owned, Esoterics and LabCorp. He also phoned me 2 years later from his phone and said, I'd show you a picture, but you're on your desk phone. I can't show you, but thanks, Russ. I have now retired because I took 15% commission for all those studies. I'm on my yacht in the bay. I'm like, Steve, you had to phone me up to say that? Thanks, mate. So I also realized there is a great career and good money, but the sales part of that guy made an absolute killing and retired early. But yeah, it forced me to actually really clean up my act and really think hard about what it means to be selective and sensitive. That was a really fun challenge. Took about a year. It was a really fun challenge.
And it sounds like the thing Steve decided to be brilliant at was sales.
Using me, yep, fair play.
Russ.
And the team as well. Andy Wagner,
Sure.
Andrew Wagner, who worked for me at the time, was also instrumental in that, as well as Mary Moore. But yeah, and Andy's gone on to great things at Bristol. So yeah. Which is another thing I think we haven't really talked about people, but I think we as scientific leaders, again, I want to echo. I think Russ gave some wonderful, thoughtful commentary in his work and discussions with you. I would fully agree with some of his sentiments. We are enablers as leaders for our people's journey. If we get them for a long time or a short time, it doesn't matter. We're trying to make the best of them, open doors of opportunity, be somewhat custodians to the best of our ability of opportunities and align them with where they want to go, but they drive their own career. And that is a mantra that I really do try and get across. It's your career, it's not mine. I'll do whatever I can to match your aspirations and your career with what we do and where you're going. But giving back, I think just giving back. As I said earlier, if we leave this industry with things in our head that we've not, transposed through writing or to somebody else's, thinking, then we've not done ourselves a service. We've done ourselves a disservice.
Yeah, it's interesting. The people I've mentored over the years, and it's always a pleasure. And you watch some of them, and they don't go anywhere, and that's okay. So I hope they're happy. I hope everybody finds their own happiness. You watch other people, and they're moving along nicely, and then you see other people who just explode and blow past what you do or what I might do or I see, but it's just it's amazing and it's gratifying in all different ways. But without a doubt, when you really have somebody who takes it on, who absorbs it, who blows past what you're even doing, whatever they are choosing to be brilliant at, maybe you've given them a kickstart in science or in leadership or whatever that is, that is such a fantastic feeling. It's probably a better feeling than any of the promotions or any of the other career advancements personally, right? It's just, at least that kind of fills me up.
I fully agree. Yeah. I think two things that I'd like to sort of annotate with that there, Chad, and that is amplification of what you are as a person, as a leader through your team is really a goal of good leadership. But stepping out the way and giving them the sunlight and not having the shadow is really key for me. And that's hard. I know I've got a relatively big mouth and a big reputation, but I actually force myself to not know things that some of my team members, my direct reports, are working on. I don't want to actually have the shadow. I want them to get all of the sunlight. I force myself to be completely removed from that, perhaps at the 10,000 foot level, so I never get looked on as the expert. Not that I don't think I could perhaps apply myself, but I need to leave sunlight room. And secondly, as you rightly say, when you've worked with a scientist and you've been part of their journey, and then you see them up on stage or in an engagement setting just knock it out of the park, I'm so proud that I've had a piece of their journey. But they've brought a lot of moxie. They've brought a lot of diligence. And it's kind of the hardest thing to capture in an interview setting and to learn, and that is drive. But to see a driven, hardworking, smart person just blow past you, some you've given opportunity, and even if you haven't, smarter than me, go for it. I want you to, your time, your journey. I think that the personal pride that I personally see when I have my team members present at big national, international meetings and just kill it. I'm like, I'm so proud.
I've got
So
all the
proud.
time in the world for those people. All the
Yeah.
time in the world. Yeah, yeah. I'm
Well,
speaking. Yeah. Oh, go ahead.
but even saying that, I had somebody recently who joined me in about 8 months, and he said, you know what, I'm enjoying the science, but I enjoy the instruments more. Okay. Well, let's get you a bit more instrument training and let's get you with the vendor. And
Hmm.
he now
Yeah,
works for the vendor and is installing a new
yeah,
mass spectrometer for me in 2 days' time. Well, okay, we've got a little bit of the journey, but my network enables you to do what you want to do.
yeah,
You want to be a
yeah,
maintenance engineer. Perfect. Let's make that
yeah,
happen.
yeah. That's a fun job. I mean, as you said, you started out breaking instruments for 6 months and then fixing them for 6 months. So yeah, yeah.
Not not all of them. Not all of them. I'm afraid the MS9 was a 60-year-old instrument that I couldn't ever fix. But it
Mm.
became. We left the electromagnet on, and it became festooned with metal parts over the next two
Yeah,
years as a
yeah.
stress toy.
Yeah, I feel very fortunate also to have started out on a sector instrument. So
Yeah,
that's
yeah.
not many people have that opportunity these days. So yeah, you were making me think actually, and I was writing down. I needed a quote from you because on Monday I'm speaking at PitCon with the Chinese American Chromatography Association, and it's a career development seminar. So I'm gonna bring up a series of quotes and that things that I've heard from people over the years that have really made a difference and made an impact. And it's amazing when you sit down and you think about the little points where different people have said something and you took something away from it. And so for about 6 months, I've just been writing these down as they pop into your head. So I think the be brilliant. It's gonna go into the talks.
Nice, nice. Yeah, the
Phenomenal.
But I
two
think
quotes from
be brilliant.
today that I'm sorry.
Yeah.
The two quotes that really impacted me, Russ, that you said were every sample is a person,
Absolutely.
and then every tube is a question that's
Yep.
asking, am I okay?
Yep.
And that really, really humanizes, the work that you do and how important it is. Because maybe every once in a while you get to see a face and you get to meet a patient, but to the vast majority of these thousands of samples, these are thousands of people that are wondering, "Am I okay?" And I think that was brilliant and heartwarming as well.
Yeah.
Thank you. I'm actually choking up a little bit right now, just even
Mm,
thinking about that, because it is the compass point that we are honoured to have the opportunity to impact positively every day. It's a true honour um,
Hmm.
to be given that solemn responsibility, and
Yeah.
we do not take it for granted. Pun intended with my surname.
Yeah. Yeah.
perfect, perfect. So Russ, what do you do? What are your favorite things to do in your free time when you're not
I
running
play
samples?
guitar in a yeah. I play guitar in a band.
Oh, nice.
We play out occasionally. We were called the Occasionals, but somebody else had that name, so the Occasional Imposters. So I play music. You probably see. In fact, I think some people may have seen me at ASMS in San Diego a few years ago. We were busking, and people sort of walked up and looked at me, looked away, and looked back again like a Tom and Jerry cartoon. They're like, "Russ, are you not getting paid enough?" I'm like, "Of course I am. I'm just having some fun." So, yeah, I play music in a band, and we actually have recorded some material. We haven't finalized the album, but we wrote a lot of our own
Nice.
stuff. A couple of years ago, we perform out. The band's called the Occasional Imposters. Don't look for us. We've not put anything. There's probably
Occasional
some
imposters.
videos on the internet of us right now. But yeah, I do that.
Sure.
I
That's
hit the
awesome.
gym. I hit the gym now 5 days a week. We're meeting at 5:15 every night, 4 or 5 nights a week for our engagement time to just decompress and have time together as our kids are now have gone to college, and my daughter has graduated and she's looking to go back to PA. So yeah, gym. I like playing golf, but I find it's a little bit too many hours of the day, and
Yeah.
it's one of those things you can't hide a bad golf swing. It's all on you. So I don't play enough to be great. I was good at one point, but
You
not
were
that
good.
good.
You were good back in the day. Yeah.
Yeah, we
Yeah.
we played together at Land
Hmm.
O'Lakes a
Few
few
times.
times.
Few times. Yeah. Yeah.
Yeah,
So is it
yeah. So
is it lead guitar? Is it acoustic? What
funny story.
rhythm?
So I got given an electric guitar for Christmas from my wife. After 9 hours of murdering "Wish You Were Here" solo with the electric guitar, she said, "I should have got you an acoustic." So within a week, I had an acoustic guitar, just so I wasn't
Nice.
torturing these wonderful songs. I play mostly acoustic. I have a beautiful D35 Martin. It was built 1970, the year I was born. that I play. It's just stunning. So acoustic guitar mostly. I got a couple of nice electrics. I'm struggling with that musician's problem. How many guitars do you need? Just one more. And my wife said, but you said that last time. I said, and I'll say it next time as well. So don't ask the question. But definitely acoustic. Lead if we got the second guitar playing, but if not, I play a lot of bass rhythm with a mandolin and fiddle and a few other things. Yeah, it's great fun. Music is just an expression of notation and mathematics. And I was a classical trained pianist as a kid, so yeah, music is a wonderful expression.
Oh, that's good. That's good. Yeah, I self-taught acoustic guitar, and I dabble in electric. I was in a band called Amendment 21 for a number of years, and we're doing our farewell concert next month because one of my friends is retiring and hitting the road. So this will be our last show.
So here's the thing. So we're called the Occasional Imposters, or was the Occasional, because we would meet once or twice a year, and occasionally there was enough of us close enough together to play. So that was where the sort of name. But you might want to do. So we played in America and in Europe. So we had a shtick of world tour. And I said, why don't we have world tour and just today's date, whatever that gig is. There's only one date on the world tour. So you might want to do something like that. He said, world tour, and it's got one date, and it's that world tour and final tour date. And it's just got one date on the back. I think there's a merch angle there. I think that'll be fun.
That's it. Brilliant. Absolutely is. That's great.
I hope you enjoy it. I certainly love expressing myself through the guitar. There's one within reach. Every time I'm at home, it's only 6-7 feet away. I play it all the time.
Well, Russ, where can people find you? How can they connect with you?
Well, I'm in North Carolina. Most people will see me at conferences like ASMS. MSACL is another conference. I'm actually really thrilled. I'm getting a Distinguished Contribution Award next month. I never win anything, guys. So I'm kind of looking forward to getting that. But an MSACL conference is one I'll be at. I'll be at the AECC or the ADLM clinical conference. So I do 3 big conferences. They can always contact me at grant@labcorp.com. If they see me at ASMS and they want to buy a Guinness, they can certainly buy me a Guinness, and I'll stand next to them as they continue to buy me Guinnesses and ask me questions. I'm happy to do that. But yeah, via email and or ASMS, MSACL, ADLM, which was the AACC meeting. I teach at those meetings, or I present at those meetings, or I'm generally there.
Awesome.
Brilliant. Thank you so much for joining us today. It was an honor and a pleasure to meet you and to chat with you.
Well, thank you, gents. Humbling to be included and asked to present. I'm kind of intrigued. So I saw Russ's 2, and then you said this is number 7. So who's 5 and 6? Who's that?
Stephanie Passes Farmer.
Who was 1 and 2 then? That was you guys, wasn't it? Okay.
Yeah.
Got it.
Yeah.
Just us getting started.
Got it. Mm-hmm.
Yeah.
Chad, thank you for the tap on the shoulder. Gregory, thank you very much for the question and answer. I hope this came across. Even we had some audio issues. I hope this came across well. And to the listeners out there, do great things, be brilliant, and always listen to your inner voice to tell you to do the right thing. But bring the passion. It's all about passion. We give up all that time with our loved ones to do this on a daily basis. Make it matter.
Make it matter. Absolutely. That's so good.